Okay, so today we will learn the chapter 20. The chapter 20 title is the Reconstructing and Using Phylogenies. Chapter 20 is composed of 4 subjects starting from the evolutionary history. Finally, we will learn the phylogeny. Okay. So this is the first subject. All of life is connected through its evolutionary history. So what is phylogeny? Phylogeny is evolutionary history of a group of organisms. In Korean, phylogeny we call it as a 계통. Phylogeny is evolutionary history. Okay, but we cannot see the phylogeny in the eye, so we made the phylogenetic tree. Phylogenetic tree in Korean we call it as a 계통수. So phylogenetic tree describes the evolutionary relatedness between the species. For example, plants and fungi, they are very close to each other. However, the plant and the cyanobacteria, they are far away from each other. So based on the phylogenetic tree, we can describe the evolutionary relatedness between the species. Okay. So in this slide, I will explain the component of phylogenetic tree. First of all, we have to learn the lineage. Kim Seo-jin, what is lineage? 게임 안 해? So lineage is a series of ancestor and descendant population. So this dot is the species, population, or gene at one point in a time. At one point in a time. But lineage means that this becomes lineage as we follow its descendants through the time. This is time through the time. So there was one species at one point in time. After that, second generation, third generation, fourth generation, fifth generation, sixth generation. So this is, we call it as the lineage. Lineage means that series of ancestor and descendant population. Okay, so this is time as times go by. Okay, and we also have to know some important terminology. Node. Okay, this is node. In Korean, we call it as 가지. So node means that point at which ancestral lineage divides into two descendant lineage. Okay, so one ancestral lineage is divided to two descendant lineage. So at this point, we call it as node. Okay, this one becomes one descendant lineage, and this one becomes another descendant lineage. Okay, and also we in the phylogenetic tree we can see the dot like this. Okay, so each lineage continues to evolve independently as different traits arise. For example, this trait and this trait. Okay, for example, this is the dog. Okay, so initially the original ancestor of dog is wolf, starting from the wolf, but one becomes like this and one becomes like this. Okay, so one became one lineage, and the other becomes another lineage. Okay, so each dot means that the different trait. So this has a bulldog, and this is another dog is like this. Okay, and also another important thing is lineage continues to split. Okay, one time they split it, and another time they also split it, and they also split it. It's like this. And next generation, maybe they will also split, and split is like this. So lineage is continuing to split. Okay, so this is the component of phylogenetic tree. The important thing is that we have to know lineage and node. Okay, and also this red dot means that different trait is like this. Okay, this is bulldog, and another dog they have a different trait. It's like this. Now, in this slide, we will learn another important terminology is the clade. Clade, in Korean, we call it as bungigun. Clade. Okay, clade means that a group consisting of all descendants from common ancestor. They have some common ancestor, and they have the all descendants. Chimpanzee, human, gorilla, orangutan is like this. So this one we call it as clade. Starting from common ancestor, and also they include all descendants. So this is we call it as a clade. In Korean, we call it as bungigun. Okay, so each tree, they have each branch. For example, this branch can be rotated like this. Initially, the chimpanzee, human, gorilla, orangutan. After rotated this branch, it will become the human or chimpanzee, gorilla, orangutan is like this. However, even though we rotated the branch, there is no changing of the meaning of the tree. So this is, we call it as the clades. Okay? Another one, we call it as a taxon. Taxon. In Korean, we call it as a 분류군. So taxon means that so this is the one clade, they have common ancestor, and they have the all of component from lamprey, perch, salamander, lizard. Finally, they have chimpanzees like this. Okay. However, this one we call it as a taxon. Okay. Mouse and chimpanzee is belong to the mammal. And next, lizard, crocodile, pigeon, mouse, and chimpanzee, they are belong to amniotes. So amniotes have the animals with self-contained egg. Okay, and next, taxon is like this. They include salamander, lizard, crocodile, pigeon, mouse, chimpanzee. This one we call it as tetrapods. So they have the four limbs. For example, human has 2 legs and 2 arms. This is 1, 2, 3, 4 limbs like this. So they are belong to the tetrapods. So finally, there is another taxon. It's included lamprey, perch, salamander, lizard, crocodile, pigeon, mouse, chimpanzee. They belong to the vertebrate. Vertebrate means that animals they have the backbone. Okay, we have backbone like this. So this is, we call it as the taxon. Okay. Initially, this one was one clade. They have the common ancestor, and they have include all descendants. Okay. Inside the clades, there is several taxon, starting from the mammals, amniotes, and tetrapods, and vertebrate. It's like this. Okay, now we have to know some important terminology: ancestral trait and derived trait. Okay, so for example, this one was ancestor. Okay, they have common ancestor, and they divided to shark and ray-finned fish, amphibians, and primate, and rodent, crocodile, and birds is like this. Okay, so this one, vertebrate means that they have a backbone. So this is, we call it as ancestral trait. The trait that is present in the ancestor of a group. So the common ancestor have the vertebrate. Vertebrate means that they have the backbone. So this one we call it as ancestral trait. Another one, for example, bony skeleton, limbs, amniotic eggs, hair, and egg and shells. We call it as derived trait. So trait in a descendant that differs from ancestral trait. Okay, so for example, they have ancestral trait is here. Okay, so ray-finned fish, they are different from ancestor. For example, bony skeleton, they are different from the difference between the ray-finned fish and the ancestor is the bony skeleton. How about the difference between the amphibian and the ancestor is limbs? How about the difference between primates and the ancestor? They are amniotic eggs, is like this. So derived trait means that trait in a descendant that differs from ancestral trait. We call it as derived trait, is like this. Another one is the synapomorphism. So the full name is shared derived trait. In Korean, we call it as a 공유 파생형질. Okay. So synapomorphism is the derived traits that are shared among a group of organisms. For example, the synapomorphism between the crocodile and birds is eggs with shell. This is the synapomorphism between the crocodile and birds. How about between the primates and the rodent? The synapomorphism is hair. It is like this. This is, we call it as synapomorphism. The full name is shared derived trait. In Korean, we call it as 공유 파생형질. Okay. Okay, now we cover some important terminology: convergent evolution. Convergent evolution. In Korean, we call it as the 수렴진화. I will explain the convergent evolution again. So this is the bat wings is like this, and this is the bird's wing. So bat wing is their composed. This is skin. Their skin. They are composed of skin. However, bird's wing, they are composed of feather. They are different. So the skin and feather, the independently evolved trait subject to similar selection pressure. What is the selection pressure? They must fly to avoid attack from predator. They have to fly. But one is the they have independently evolved traits. One is skin, one is evolved. Okay, but they are subjected to similar selection pressure. They have to fly. But this trait becomes superficially similar. They becomes wing. They becomes wing. This is a bat wing, and this is the bird's wing. They become superficially similar. So this one we call it as convergent evolution. Another example is that this is shark in underwater, and this is dolphin, and this is the ichthyosaur. This is the 공룡. Okay. So this is fish, and this is belong to reptile, and this is belong to mammal. Because but they have the similar selection pressure. Okay, they have to adapt. They have to survive under the water. Under the water. Okay, that's why the fish and reptile and mammal, they have the similar body shape is like this. So this is, we call it as another example of convergent trait, convergent evolution. Okay, now we cover analogous character and what is the homologous character. So previously, maybe in high school, we didn't cover convergent evolution. We only cover the analogous character and homologous character. Let me see some details. So analogous character, in Korean, we call it as 상사형질. And homologous character in Korean, we call it as 상동형질. Okay, so what is analogous character? They are functionally similar structure. They have a wing. They are wing, but they have independent origin. For example, bat wing comes from the skin, and wing of birds comes from feather. So this one, we call it as the analogous character. Another one is we call it as a homologous character. For example, this is the human's arm. This is cat the leg. And this is whale's flipper. And this is bat wing. They are functionally different structure, but they have same origin is like this. They have same origin like this. They have same bone, it's like this. Their bone structure is same, like this. So this is example of arm of human, leg of cat, flipper of whale, and wing of bat. Their origin is same. So this is, we call it as humerus, and this is, we call it as a radius. Another bone, we call it as a ulna, and this is the carpel, like this, okay. Okay, so this is, we call it as the homologous character, okay. Okay, so now, once again, so in first subject, we cover very important terminology, lineage and node, okay? And also, each spot means that different traits. Okay, we also cover clade, what is the taxon. And also we cover some important terminology, ancestral trait and derived traits. And also shared derived trait, we call it as a synapomorphism. And we cover convergent an analogous character and homologous character. So this is the first subject. Now we will go to the second subject. So phylogeny can be reconstructed from traits of the organism. So in this slide, we will show how to construct a phylogenetic tree. Okay? First of all, we have to assign in-group and out-group. So in-group is the primary interest. Okay? So for example, people is interested in the in-group, including human and chimpanzee and mouse. So this is a very, the primary interest group, we call it as in-group. And the remained group, we call it this one, lamprey. In Korean, we call it lamprey, means that we call it as the 칠성장어. So no one is interested in the lamprey, so it becomes out group. Okay, so there we first of all compose split into in group and out group. Okay, in group and out group, they have the common root of tree. This is root of tree. So root of tree is, this is the root of tree is located between the in group and the out group. Okay? And second, we have to mark a synapomorphism, okay? Between the chimpanzee and mouse, they have the synapomorphism. This is a mammary glands. Okay, so mouse or female mouse has also mammary glands, and chimpanzee also has mammary glands. It's like this. For example, pigeon, crocodile and pigeon. Okay, they have the zizad. This is zizad. In Korean, we call it as 모래주머니. Pigeon has zizad. They are composed of lots of sand. And also crocodile. They also have this kind of organ, zizad. Okay, so this zizad becomes the synapomorphism. Okay, so usually each clade, this is one clade, so each clade shares at least one synapomorphism. So in this clade, there are lots of the synapomorphisms. It's like this. And also, explain the observed data very simply. We call it as a parsimony principle. Parsimony principle, in Korean, we call it as the 전략 법칙. Okay. Okay, for example, I will show you some example. Okay, so lamprey, we call it as the 칠성상어. If we compare the lamprey and chimpanzee, okay, their morphology is totally different. But when we explain the difference between the lamprey and chimpanzee, we always explain very simply. For example, chimpanzee has jaws, but lamprey, they don't have jaws. So this is, we call it as, we follow the parsimony's principle. Whenever we explain the difference between the lamprey and the chimpanzee, there are lots of difference, but we cannot explain it in details. Just simply, the main difference between the lamprey and chimpanzee is lamprey, they don't have jaws, but chimpanzee, they have jaws like this. Okay, so this is, we call it as parsimony principle. 장성인 알겠어? Wake up. Okay. And also, so this is the we learned how to construct phylogenetic trees, and also in this slide we will see some important concept. Two important concept is used to construct phylogeny. First of all, morphology. Second of all, developmental pattern. Okay. First, let me start from the how to construct the phylogeny. We always based on the morphology. So similarities in morphology reveals evolutionary relationship. So this is the bone structure. For example, this animal and this animal, their skeletal system is nearly similar. So it means that they are evolutionarily very close. However, this animal and this animal, their bone structure is different. That's why they are evolutionarily, they are far away from each other. Okay, so this is one example. And another example is the floral structure in plants. For example, this flower and this flower, they are close to each other. However, this flower and this flower, they are far away from each other. So whenever we construct the phylogeny, we always, first of all, we always use the morphology. Okay. Another thing is we also have to consider developmental pattern. Okay, so this one is, we call it, this is one example. This is adult. This is a C skirt. In Korean, we call it as a 바다물총. So in the bottom of C, they did not move. Okay, they attach the rocks in the bottom. We call it as the C skirt. And this is a frog. Okay, their morphology is totally different. So based on the morphology, their evolutionary relatedness is far away from each other. However, when we see their developmental pattern, for example, in the early stages, this is the sea skirt larvae, and this is a frog larvae. They are nearly similar to each other. So they also have the neural tuber and notochord. Frog also has neural tuber and notochord is like this. Okay, of their morphology is totally different. However, the developmental pattern, they are nearly similar to each other. So what is notochord? Notochord becomes the vertebrate column is like this. Okay, so usually the similarity in developmental patterns reveals evolutionary relationship. For example, sea skirt and vertebrate have the notochord at some times in their development. So morphology, based on morphology, for example, based on morphology, this is A, B. Maybe they are far away from each other. Maybe this is the C is like this. This one becomes the sea skirt. This one becomes the frogs, it's like this. However, developmental pattern, they are close to each other. This is the sea skirt, and this is the prog. It's like this. So we have to consider both of them, morphology, and also we have to consider developmental pattern. Each other. Okay. Okay, so in this slide, we will learn how to test accuracy of phylogenetic analysis. Okay, so based on so we are very curious about, we construct phylogeny based on the morphology, and also we construct phylogeny based on developmental pattern. So our question is the phylogeny tree is accurate or not. Okay, this is, I will show you one example. So in this slide and next slide, we tested the accuracy of phylogenetic analysis. We tested the accuracy of the phylogenetic trees. Okay, so this figure is very difficult to understand it, but I make the very simply explain to you, okay? Even though you read the textbook, you cannot understand it. But I did it. I did it, okay? So this is recorded as a viral plug. So in this colony, there are lots of the virus is located here. This is recorded as a viral clock. Okay, there are several colonies, but we only can select one single viral block. So this one will become the common ancestor. This one will become the common ancestor. Okay, after that we added a lot of mutagen. Mutagen were added into the viral culture to increase mutation rate. For example, we can add some radiation, or we can add some chemical in the virus culture. Also, we can add some infectious agent to the viral culture, so it will increase the mutation rate. Okay. Okay. After that, initial culture was split into the two separate lineages. One becomes in group, and the other becomes out group. Okay, and also split each lineage every 400 generation. Every 400 generation. Okay, split and every 400 generation. First generation, second generation, third generation, or fifth generation, 400 generation later, they will split it. After that, first, second, third, fourth, that 400 generation split it is like this. Okay, so after that, we will make the outgroup lineage in the A lineage, D lineage, C lineage, E lineage, F lineage, H lineage, B lineage, G lineage. Okay. So, okay. At the time of the split, the gene of each ancestor was sequenced. And also, the gene of the end points of lineage were sequenced. Okay. After that, we got a lot of data, in data. Okay. After that, we just give it to this kind of data to another people. Okay. So we have all of data, but we did not analyze the phylogeny tree. So we give all of the data to another people. And another people, they did they used the DNA sequence. So they did phylogenetic analysis, and finally they will make some kind of the phylogenetic tree is like this. Okay, but people found that this phylogenetic tree and this phylogenetic tree, they are same. So based on this result, people say that the accuracy of phylogenetic analysis is very, very higher. Okay? This is the one example of to show that the accuracy of the phylogenetic analysis. Okay? So phylogenetic analysis of DNA sequence accurately reconstruct evolutionary history is like this. So they exactly make this kind of phylogenetic tree is like this. Okay, I try to explain very simply. Very difficult to understand. I know, very difficult to understand it. Okay, I try to make it very simply. Okay. And also, now we will go to the third subject. Phylogenies makes biology comparative and predictive. Okay, so phylogenetic trees can be used to reconstruct past events. Okay, for example, zoonotic disease. So this is recorded as zoonotic disease, transmitted from animal to human. So this is recorded as the zoonotic disease. For zoonotic disease, it is very important to understand where, how, and when it enters the human. So now people think that maybe COVID-19 comes from this is the Korea and this is the Asia, China. Nowadays people thought that COVID-19 comes from some China, for example, Wuhan. Okay, people predicted that like this. So whenever we predict this one, we always use phylogenetic trees like this. Okay, so for example, people make phylogenetic trees is like this. Okay, okay, they have common ancestor, and in human we found HIV1. And also some chimpanzee, we also find SIV CPZ virus. And also monkey, we find the SIV host virus. And the monkey, another sun-tailed monkey, we have found SIV sun virus is like this. First of all, we made the phylogenetic tree is like this. So based on these phylogenetic trees, people found that HIV1 occurs from the chimpanzee. Okay, and also HIV2 comes from Suti mangabees. In Korean, we call it as Ginkgori 원숭이. They have long tail is like this. Okay, so based on this phylogenetic tree, we can reconstruct past events. Okay, maybe HIV1 comes from chimpanzee, and HIV2 comes from the Suti mangabey is like this. Nowadays, Chinese people say that COVID-19 comes from their country. But nowadays people making the phylogenetic trees, maybe the WHO will know soon the origin of COVID-19. So they will use the last of phylogenetic tree. So they will know soon. Maybe the COVID-19 comes from China, I think. Okay, now we also cover very difficult to understand concept, molecular clock. So we have clock. Now we will cover molecular clock. In Korean, we call it as 분자시계. What is molecular clock? I will explain it. So this is the graph. X-axis is composed of the year, and this is Y-axis is the proportion of amino acid difference. So we have this kind of slope. Okay, slope means in Korean we call it as 기울기. The slope represents an average rate of changes in amino acid sequence. Okay, so based on this molecular clock, we made molecular clock hypothesis. So previously we learned that mutations accumulate in continuous fashion over time is like this. So mutation occurs continuously is like this. Okay, so rate of the molecular changes, usually they are very constant, is like this. That's why we can predict the timing of evolutionary divergence. For example, 100 million later, there will be 0.2 amino acid difference will be happens. For example, 200 million later, maybe 0.35 amino acid difference. And also 300 million later, maybe the 0.55 amino acid will be difference. 400 million later, maybe 0.8, or 500 million later, maybe 0.9 amino acid will be different. It's like this. 장성현, wake up. Okay? So this is the molecular clock hypothesis. Because this rate is constant, based on this slope, we can predict the timing of evolutionary divergence. Okay, 100 years later, there will be some difference, and we can predict it 500 million later, there will be like this. Okay. So this is how to date the origin of HIV-1 in human population. Okay, this is very difficult to understand it, but I will make it very simply, okay? So HIV, we don't know when it entered to human. We don't know, okay? But nowadays people make a lot of phylogenetic trees. Now that we predicted that. I will show you how they did it. Okay. So this is the phylogenetic trees. The tree of HIV samples plotted on a divergent axis. So this sample we can collect in 1990. This sample 1997, 1983, and 1984, and this sample 1959 is like this. They have the common ancestor of HIV is like this. Okay. This is the phylogenetic tree of samples of HIV virus. Okay. After that, so based on the sample taken between the 1980s and 1990s, So we made the graph is like this. Starting from the 1980 and 200, 2000. So this is the years, and this is the brain more the difference from the common ancestor is like we made the graph is like this. So this is this graph is belong to this one. So based on this graph, we can make the graph is like this. Okay. Now we just tested this molecular clock. Okay, so for example here we can see that this sample we collected is 1959. So this molecular clock was tested using samples from 1950s. Okay, so based on this molecular clock, so this 1959 sample was predicted the 1957 plus minus 10 years. Okay, so it means that this molecular clock is accurate. Okay, so we had this sample. This sample comes from 1959. Okay, but we give this information. They have some kind of information. So we give this information to another person. So another person found that the divergence is 0.06. Okay, so based on this graph, they predicted that this sample comes from the 1957 plus minus 10 years. They are nearly similar to each other. Based on this one, we can say that molecular clock is accurate. Okay, so based on this graph, we estimate that HIV-1 comes from 1920, 30, something like that. So based on this one, molecular clock estimated that HIV-1 enters human in 1930s. In Africa, in Africa, there are lots of people just eat the chimpanzee. So they kill the chimpanzee. So they directly eat the chimpanzee's brain. So maybe at that time, the HIV-1 is entered to the human. So that date may be the 1930s. So based on this molecular clock, we can estimate that the HIV-1 is entered in 1930s. Okay, so this is very difficult to understand it. I know, but I try to make it very simply. Okay. Now we will go to the fourth subject. Phylogeny is the basis of biological classification. Now we have to know what is the monophyletic group, and also we have to know what is paraphyletic group, and also we have to know what is polyphyletic groups. Let me start from the monophyletic group. So this one, they have for example, they have common ancestor. This blue box, we call it as a monophyletic group. Okay. This blue box is one taxon. They are composed of a common ancestor, and they have the Hij, they have descendant. Okay, this one, this blue box, we call it as the monophyletic group. Contains common ancestor and all descendant. Okay, so this group can be cutted from the tree with a single cut. With a single cut, this can be removed from the original tree. It's like this. So this one, we call it as the monophyletic group. You have to know to solve the final term exam. You have to know what is the monophyletic, what is a paraphyletic, something like that, okay? So this is we call it as a monophyletic group. Monophyletic group taxon contains all of ancestor and all descendant. Another one is paraphyletic group. So they have the common ancestor, okay? But the green square, a group that does not include all descendants of a common ancestor. They did not include A. So in this case, in this group, we call it as a paraphyletic group. This one, we call it as paraphyletic group. They have common ancestor. They include B, C, D, but they did not include A. So this one, we call it as paraphyletic group. Another one is we call it as the polyphyletic group. For example, the red color, red scale, they are composed of E, F, G. They composed of E, F, G. The common ancestor of E, F, G is located here. So this is common ancestor of E, F, G. However, this scale, they did not include the common ancestor. So in this case, we call it as a polyphyletic group. Okay, once again, monophyletic group, they have the common ancestor, and also they include all of descendant. Paraphyletic, they also include the common ancestor, but they did not include all of descendant. Okay, this one, they did not include it. Polyphyletic group, they did not include the common ancestor. This one, we call it as polyphyletic. You have to know. Okay, and finally, we will cover binomial nomenclature. Okay, whenever we did biological classification, we always use binomial nomenclature. Okay, for example, human, we call it as Homo sapiens. Homo sapiens. Okay, this is the genus, and this is species. It's like this. So we always use the binomial nomenclature. We include the genus and species. For example, so every species have a unique scientific name that consists of two parts, genus and species. Okay, human, we call it as a Homo sapiens. So, for example, this is another example, the lion. The common name, we call it as lion. Common name, we call it as human. But their scientific name is their genus is Patera, and their species is Leo, is like this. So we always use the binomial nomenclature. Okay, whenever we use the biological classification, we always use binomial nomenclature.